Hi all, in this video we are going to study about the physiology of the digestive system. So whenever we are looking at the digestive system, we look at different parts of the digestive system and we start with the processes that takes place in mouth. Okay, so these are the various processes that you see over here, the three that is listed over here: salivation, mastication and deglutition. So these are the three processes that we need to study under the mouth. Our mouth is otherwise referred to as the buccal cavity. Okay, so whenever we look at any part of the GI tract like stomach or intestines or large intestine, we have to know about the secretions of that part. So what are the secretions that takes place? Okay, for example, in the mouth the secretion is the saliva. Okay, and also we have to know about the movements that takes place. Okay, the movements that takes place in that particular part. Okay, so in the mouth there are two different types of movements that takes place. One is the mixing movements that is happening, is called as the mastication, and the forward movement of the food particle, that is called as the deglutition. Okay, and as far as the physiology is concerned, the regulation of these, regulation of the secretion, the regulation of the movements. Okay, so that is a regulation. There are two different types of regulation: hormonal regulation and neuronal regulation, that is the sympathetic or the parasympathetic regulation. Okay, whereas in the case of like salivation, we know that it is mainly regulated by the neurons, especially by the parasympathetic neuron regulate the secretion of the saliva. Okay, so as far as physiology is concerned, the regulation part is very important for the exam point of view. Like you get the most of the questions from the physiology with regarding to regulation. So please make sure whenever we discuss the regulation, take that as a high yield point and concentrate on the regulation of secretions, movements of any part of the GI tract. Okay, so if we start with the first one, that is the salivation. Okay, salivation, as you know, it is happening from the salivary glands. Okay, so what is the basic role of salivation? So why we need to do the salivation? What are the important roles played by the salivation that is being described over here? So the major one, it helps the swallowing process or the deglutition process, and they keep the mouth moist, not allowing it to dry it out. Okay, because these are mucous membranes, we need to keep it moist, and act as a solvent, for the molecule to stimulate the taste buds. Okay, so you know what are the different tastes that we have, like sweet, salt, sour, bitter. Okay, so these are the various tastes that we have. Like sweet is by the glucose molecule, and the salt is by the sodium chloride, and the sourness is mainly caused by the acidic substances mainly causes the sourness. Bitter is by the alkaloids, the bitter taste is caused by the alkaloids. Okay, and also like there is like additional taste that we have, like one more like additional taste that we have, that is called as umami taste. Okay, umami taste, that is caused by the Ajinomoto or the monosodium glutamate. The monosodium glutamate is the glutamate that produces the umami taste. Okay, so these are the various different types of taste. That, like for that you need the it has to be dissolved. This taste molecule has to be dissolved so that it can very easily stimulate the taste buds. Okay, so the saliva act as a medium to dissolve all these molecules in order to be like easily interacting with the taste receptors that is present on the tongue. Okay, and next thing is like it aids the speech by facilitating the movements of the lips and the tongue. Okay, the lips and the tongue. So the lubrication that is provided by the saliva helps for that. And the next function is it keeps the antibacterial action that helps the mouth and the teeth clean. Okay, in case if you don't have the saliva or decreased saliva, condition called as what, xerostomia, so the person will end up in damaging the teeth, because it's essential to keep the teeth clean. You need the saliva. So what are those substances? We are going to list slightly later those substances that helps for preventing the decay of the teeth and how it brings about the antibacterial action. So we are going to list all the substances that helps for that. The buffers in saliva, okay, to help them maintain the pH of about 7. Okay, it's like slightly acidic pH. Okay, so the normal pH of the plasma is about like 7.35 to 7.45. Okay, that is for the plasma. So this is your normal pH, whereas you have a slightly acidic pH for the saliva. Okay, slightly acidic pH. Okay, and they also help neutralize the gastric acid and relieve the heartburn when gastric juice is regurgitated into the esophagus. Okay, so later on we will study the esophagus. If the acid comes back, we call it as the GERD, the GERD. Okay, gas esophageal reflux disease. So during these conditions, when the acid is refluxly coming back to the esophagus, to protect that we need saliva. Saliva helps to protect that for the neutralization purpose. Okay, now we will see little bit about the histology of the salivary glands. Okay, so the salivary glands that is shown over here is mixed type. Mixed type means it has two different types of acini. Okay, the two different types of acini, they are the mucus secreting acini, and the serous acini. Okay, the serous acini. Okay, so the serous acini is shown over here. This is the serous acini that you can see over here. Okay, the serous acinus. Okay, the singular and the plural for acini and acinus. Okay, and the another one, that is the mucus acinus. Histologically, you can see the differences over here. This nucleus is more like towards the center. Okay, the nucleus is more towards, and you can see a lot of like secretory particles, like secretory vesicles over here, and also you can see that the nucleus is little elongated over here for the mucus secreting glands, and it's towards the basolateral side. Okay, this is the basal side, and this is the apical side of the gland. Okay, this is the apical, this is the basal. Okay, so when you take a cell, okay, so this is, this is your cell, and this is your lumen. Okay, this is your lumen. Okay, so this side over here, this whole part is referred to as the basolateral side. Okay, and this is the luminal side. Okay, so the secretory vesicles, like, and the other particles that you need to secrete is secreted to the lumen, and the major pump that works over here on the basolateral membrane is your sodium potassium pump, which will pump the sodium out and the potassium in. Okay, and the potassium in. So this is the sodium potassium pump that works, and all other, this is the primary pump that we have that uses the ATP. Okay, and that is present on the basolateral membrane, and on the luminal membrane you have the other secretory vesicles and the other channels that help to secrete the sodium, the chloride, and the potassium, etc. Okay, so these are like the important acini we have, and because it's a mixed one, on top of the mucus secreting acinus itself, you can see a demilune. Okay, that is secretory for serous. Okay, serous secretions includes mainly the water and electrolytes, water plus electrolytes. Okay, the major one, and also it includes the enzymes. Okay, enzymes, whereas the mucus secreting contains the mucin. Okay, this is the glycopolysaccharide that is making the saliva lubricate and make it thick. Okay, serous makes it more watery. Okay, so this is the acini, and within the acini or within the ducts you have myoepithelial cells, which when it is contracting will cause the propulsion of the saliva. Okay, so the contraction of the myoepithelial cells is helped by the neurons, and then that causes the the movement of the saliva within the acini and then to the ducts, and then it is being secreted to the mouth, to the buccal cavity. Okay, so when we have finished with the acini, and the acini secrete the saliva into the duct. So we have two different types of duct. One is the intercalated duct, and another one is the striated duct. Okay, so the intercalated duct mainly handles the H+ ions and the bicarbonate. okay, HCO3 minus, HCO3 minus. Okay, so the bicarbonate and the hydrogen ions is being handled. Okay, the reabsorption of these is done at the level of intercalated duct, whereas the striated duct modify the sodium potassium content of the sodium and the potassium content of the saliva. Okay, and the striated duct is also mainly influenced by aldosterone, the hormone aldosterone. Okay, the aldosterone influences the striated duct to change the sodium concentration and the potassium concentration of the saliva. You know that aldosterone causes what? The reabsorption of sodium and secretion of potassium. So when they reabsorb the sodium, okay, so what happens to the tonicity? The tonicity of the saliva will be decreased in the presence of aldosterone, and the potassium concentration of the saliva will be increased and the sodium concentration will be decreased in the presence of aldosterone. But importantly, remember, the secretion of the saliva is not influenced by aldosterone. Aldosterone just modifies in the duct. The secretion of the saliva is mainly controlled by the acetylcholine. Secretion is controlled by the acetylcholine from the parasympathetic. Secretion is controlled by acetylcholine. Okay, acetylcholine coming from the parasympathetic nerves. Okay, parasympathetic nerves. Okay, so that is the major regulation. Aldosterone just modifies. So mainly the salivation is mainly regulated with the help of neurons and not by the hormones. Okay, so remember, aldosterone only modifies the ducts and controls the sodium and the potassium concentration. Okay, so we have seen the histology of the salivary gland. Now let us see what are the different types of salivary glands that we have in our mouth. Okay, the different types of salivary glands that we have. Okay, so the salivary glands that we have mainly includes these three: the parotid, submandibular and the sublingual, and also some minor glands and within the oral cavity, the small ones. Okay, that contribute about only like 5%. All the remaining is like the 95% is contributed by these glands. Okay, and which is about 1.5 liter of saliva per day, which is quite a huge number. Okay, so 1.5 liters of saliva is secreted per day on an average. Okay, so the biggest gland, there is the big one. Okay, the biggest gland is the parotid. Okay, whereas you can see the percentage of the saliva secreted is only 20%. The percentage is only 20%. Okay, even though that is the biggest one. So, and that is a serous type and secretes the watery. Okay, but it includes the enzymes. The watery secretion includes the enzyme. Okay, whereas the submandibular, which is below the mandibles, or like it's sometimes referred to as submaxillary also. Okay, is a mixed gland. Okay, it's a mixed gland. That means it is both serous and mucus secreting. Okay, and that makes the moderately viscous secretion. Whereas the sublingual is a small gland and only contribute towards like 5%. Okay, mainly secrete the viscous. Whereas the major one, that is the submandibular, is the mixed one that contributes 70% of the saliva. Okay, so now what do they secrete? The serous cell secretes the taline, and the mucous cell secretes the mucin. Taline is otherwise called as the salivary amylase. Okay, salivary amylase. That's another name. Okay, which is helping for the carbohydrate digestion. Okay, so this is the enzyme that is secreted. Mucin is a glycopolysaccharide. Okay, so the salivary amylase, and another enzyme that we have in the saliva is the lipase. This is for the lipid digestion and this is for the carbohydrate digestion. Okay, so let's study about the details of these secretions like water and electrolytes and the composition of the saliva. Okay, so the composition of the saliva. Okay, so first is the pH. The pH of the saliva is slightly less than 7. Okay, so less than 7, which is slightly acidic. We have already mentioned this. Okay, it's slightly acidic. Okay, but during active secretion, when there is no time for the intercalated duct, okay, to modify the saliva, what happens? The pH approaches 8, which is alkaline pH. Okay, it becomes alkaline. When it is fast secreted, okay, or when the intercalated duct does not get sufficient time to modify. Okay, so what does the intercalated duct do? Actually add acid. Okay, this is because it is adding, the intercalated ducts add H plus ions, add the acid. So because it is adding the acid, it is becoming slightly acidic, even though you can say the 7 is neutral, but for us, because the plasma pH is only 7.4, so this is becoming more acidic over here. Okay, so the alkaline is due to the lack of time for the modification by the intercalated duct. Okay, so when it is actively being secreted. Okay, so saliva contains two digestive enzymes. They are the lingual lipase and the amylase. Okay, so lipase digests the triglyceride. Okay, the triglyceride. Lipase digests the triglyceride. Triglyceride means what? It includes a glycerol molecule which is like 3 carbon structure, the glycerol molecule. Okay, and attached to each carbon is the fatty acid. Okay, is the fatty acid. So when the lipase act on this, what they do? They cleave these two bonds. Okay, the bond over here and the bond over here. That means it makes what? Two molecules of free fatty acids, okay, and one molecule of two monoglyceride. Two monoglyceride means this is for the position. Okay, so this is the monoglyceride. You have one single fatty acid attached to the glycerol molecule, so this is called as a monoglyceride. Okay, so the position, this is 1, 2 and the 3 carbon, so this is the position, so this is called as a 2 monoglyceride. Okay, don't confuse this 2 free fatty acid, they are like number, but this 2 is here for the position. Okay, 2 monoglyceride. Okay, so that's for the lingual lipase. Okay, lingual lipase. Okay, so now let us see for the amylase. What does the amylase do? Okay, amylase breaks down the carbohydrate. The major carbohydrate that we consume is our starch. Okay, so the starch contains like glucose attached to each other. The glucose is attached like one glucose here, and the glucose here, and the glucose here, and the glucose here, glucose like this. Okay, and then they will have branching. Okay, the branch. Okay, like this. Okay, so they will have branch like this, and then they add more glucose to that. Okay, so all these bonds that are like linear is alpha 1,4 linkages, first carbon here and the fourth carbon here, whereas on the branch you have 1,6 linkages. Okay, the 1,6 linkages. Okay, so our amylase, the salivary amylase or the pancreatic amylase, both salivary amylase and the pancreatic amylase can only break 1,4 linkages. Okay, but spares cannot break 1,6 linkages, and also some few of the 1,4 linkages which is terminal and the 1,4 linkages next to the branching points cannot be broken down by the amylase. So what happens? They are breaking down the large polysaccharide into smaller particles, smaller polysaccharides. Okay, but if you give time for that, if you give like sufficient time, what will happen? It can break down into individual monosaccharides, and the individual monosaccharides can be absorbed very easily from the mouth itself. Okay, so the point is, like for example, when a person eats the dinner and maybe like the person is tired and like did not brush the teeth, so what are the things that will stay back in the mouth for the early morning? It will be only the protein. Okay, for example, if the person has taken the chicken and with some butter or like rice, the rice particles or the butter all will be digested by the lingual lipase and by the amylase. But what will stay back? The chicken particle, that is like the protein. The protein will not be digested, and that proteins will stay back because no protein digesting enzyme. Okay, so the saliva does not have the protein digesting enzyme. Okay, so no protein digestion in the mouth. Okay, that's why you can see the protein staying back in the mouth for longer duration. Okay, saliva also secrete the mucin, which are the glycoproteins. Okay, so that helps for the lubrication of the food and bind bacteria and protect the oral mucosa. So that is the role of the mucin or the mucopolysaccharide. Okay, okay, the glycoproteins. Okay, so the other things, like for the bacteria, for the protection of the bacteria, what are the other things that we have? The important one is the immunoglobulin, IgA. So maybe you know the different types of immunoglobulins. They are the G, A, M, D and E. Okay, these are the 5 different types of immunoglobulins that we have. And the A, this is the A, that is the one that is being secreted in all secretions. All secretions in our body have immunoglobulin A being secreted. All the secretions in our body have IgA. So IgA helps to neutralize the bacteria, okay, and act as bacteriostatic. Okay, prevents the growth and development of the bacteria. It stops the multiplication of the bacteria and neutralizes the bacteria. So that is the role of the IgA. The other thing that is there is lysozyme, okay, which can kill the bacteria. So this is not bacteriostatic, it will be bactericidal. Okay, it will kill. The lysozyme will kill the bacteria. Lysozyme comes from the cell organelle called as what? The lysosome. Okay, so which attacks the walls of the bacteria. Another substance is lactoferrin, okay, which hold the, ferrin means the iron, which binds the iron. Okay, so which binds to the iron and also act as a bacteriostatic. 2 important role for binding the iron. So if you bind the iron, one is it is not available for the bacteria and the bacteria cannot use that. Not only that, okay, the iron is very corrosive in nature, and when it binds to the teeth and the enamel, it can cause corrosion over there. So the lactoferrin prevents the corrosion of the teeth and prevents the decaying of the teeth. So it is useful for the protection of the teeth. But another major thing that helps for the protection of the teeth is proline rich proteins. Okay, the proline rich proteins that protect the tooth enamel and bind the toxic tannins. Tannins are the ones that we see, like that can create the blackish color, okay, the blackish or the yellowish color discoloration of the teeth. Okay, and what does the proline rich proteins do? They hold it and does not allow it to get deposited onto the tooth enamel and protect the teeth. Okay, so these are the major composition, the important contents of the saliva, and we have seen the role of this. Okay, so now let us move on to the ionic. Okay, how the ions like sodium, potassium and the chloride is regulated. Okay, and what are its importance. Okay, okay, so we have also discussed about the isotonic nature. When it is being secreted, saliva is isotonic to plasma. Okay, isotonic to plasma. Same concentration of the sodium, potassium, chloride, bicarbonate, everything is equal to that of the plasma. Okay, but what will happen? The excretory ducts. Okay, there are 2 types of excretory ducts we have already mentioned. They are the intercalated ducts and the striated duct. Okay, the intercalated duct and the striated duct, these are the excretory ducts that modify the composition of the saliva by taking out the sodium and chloride and by adding the potassium or adding the bicarbonate and also adding the H plus ion. So these are the H plus and the bicarbonate is mainly regulated by the intercalated duct, whereas the striated duct mainly influences the sodium and the potassium concentration. Okay, so the ducts are relatively impermeable to water. So at low salivary flow, when there is sufficient time for the modification, ducts are relatively impermeable to water and it will reabsorb the sodium chloride, etc. So what will happen to saliva? The saliva will become hypotonic. This hypotonicity of the saliva is very important for appreciation of the taste. Okay, so the hypotonic, if the saliva is not hypotonic, the person will not feel the taste. Okay, because it's important for these food molecules, the particles in your food like the glucose or the sodium or the chloride, the salt, etc., to be dissolved into the saliva, then it can be easily sensed by the taste buds. But what happens if it is not hypotonic? Instead, if it is hypertonic, the saliva itself is hypertonic and it becomes difficult for the other particles, food particles, to get dissolved and then stimulate the the taste buds. Okay, so when you give sufficient time and a low flow, it becomes hypotonic. Okay, and then it becomes slightly acidic. Okay, that is the pH is 7 only when you have sufficient time. If there is no time, the pH also will go into alkaline, that is pH of 8. Okay, and when you have sufficient time, it will become rich in potassium but relatively depleted of sodium and the chloride. Okay, the sodium and the chloride will be decreased, well, but there will be more potassium. Okay, and that is also controlled by the aldosterone. Okay, so when saliva flow is rapid, there is less time for the ionic composition to change in the ducts. So saliva is closer to isotonic, okay, with a higher concentration of sodium and chloride. So mostly in the exam point of view, you have to remember the aldosterone, okay, increases the potassium concentration and reduces the sodium concentration of the saliva. Okay, so in a disease condition where there is deficiency of aldosterone called as the Addison's disease, what will happen to the sodium and the potassium ratio? As you can imagine, what will happen? It will become high. Okay, there will be an increase in the sodium and the potassium ratio because there is no aldosterone to reabsorb the sodium. So what will happen? There will be more sodium in the saliva. Okay, so because there is more sodium in the saliva, you will see an increased ratio of the sodium potassium. Okay, in the conditions like Addison's disease where there is deficiency of aldosterone. Okay. Okay, so now let's move on to the very important portion, that is for the regulation of the salivation. Okay, so the regulation. So what is the most important regulation of the salivation? Okay, the most important one that regulates the salivation is the parasympathetic neurons. Okay, the parasympathetic neurons. Okay, so what does the parasympathetic neuron do? Okay, let's see what are the parasympathetic neurons, how does it influence. Okay, also remember, not only parasympathetic, there is like influence of the sympathetic. Okay, sympathetic can also influence the salivary glands, not only the parasympathetic nerve supply. Okay, but what does the sympathetic do? Sympathetic causes vasoconstriction. Okay, and causes secretion of small amounts of saliva which is rich in organic constituents, that is the enzyme rich and the mucus rich. Okay, especially from the submandibular gland. Okay, so these organic constituents means it includes the enzymes and mucin. So what will happen to the saliva in the influence of, under the influence of sympathetic? The saliva will become thick and it will reduce its content because of the vasoconstriction. Okay, so how does the sympathetic cause vasoconstriction? Sympathetic causes vasoconstriction by releasing norepinephrine. The norepinephrine acts through the alpha receptors and cause the vasoconstriction. Okay, so remember that. Okay, now let us go to the most important regulation part, that is the parasympathetics. Okay, so where do we get this parasympathetic from? To the saliva. We get the parasympathetic from the cranial nerve 7 and from the 9th cranial nerve. Okay, and it takes the information from the salivary nucleus from the medulla. Okay, so the salivary nucleus of the medulla receives the information from the higher brain centers. Okay, so what are the factors that can stimulate the salivary nucleus? For example, the smell of the food, the taste of the food, sound of the food, okay, even the sight and even thought of the food. Okay, even the thought of the food, okay, can cause the higher brain centers to stimulate the salivary nucleus, whereas those things that can cause inhibition of the salivary nucleus includes the factors like sleep, and when the person is very tired or like fatigue, and fear. Okay, fear will induce sympathetic, and this fear can also inhibit the salivary nucleus, so the parasympathetic will be inhibited due to fear. Okay, so these are the factors that can inhibit the salivary nucleus. So ultimately what happens, the parasympathetic brings the preganglionic neuron. We have seen before, the parasympathetic and sympathetic both have a preganglionic neuron, and then it ends on the ganglia, and then there is a postganglionic neuron. This is the ganglia. Okay, this is your ganglia. Okay, so you can see over here in the parasympathetic you have a long preganglionic, okay, and a short postganglionic neuron. Okay, so what is the neurotransmitter released by the both the preganglionic neuron and the postganglionic neuron? Both of them release acetylcholine. But very important, what happens? This acetylcholine released by the preganglionic neuron act on the nicotinic receptor, whereas the acetylcholine that is released by the postganglionic neuron act on the muscarinic receptor. Here specifically it act on the M3 muscarinic receptor. Okay, so whereas the acetylcholine from the preganglionic act on the nicotinic receptor. Okay, so the preganglionic neuron comes to the ganglia. What is the name of the ganglia? We have two different types of ganglia. One is the otic ganglia that supplies to the parotid gland, and then we have the submandibular ganglia that supplies to the submandibular gland. Okay, so this is the submandibular gland. So the preganglionic neuron release acetylcholine. Here also the preganglionic neuron release acetylcholine. Okay, so what is the receptor for this acetylcholine? It's nicotinic receptor. Here it is the nicotinic receptor. Now the postganglionic neuron release acetylcholine. The postganglionic neuron release acetylcholine and act on the gland itself, on the parotid gland and on the submandibular gland through the M3 receptor. Okay, so remember the muscarinic receptor, M3 receptor. So under the influence of the muscarinic receptor, what will happen? There will be increased salivary secretion, especially the watery secretion. Okay, it causes especially the watery and electrolyte rich secretion. Okay, so how this acetylcholine have the influence? By the direct action on the gland, that is the SNR secretion is stimulated. Another one is to oppose the action of the sympathetic. Sympathetic is responsible to cause vasoconstriction, whereas the parasympathetic releases a substance to cause vasodilation. What is the name of that substance that can cause vasodilation? We call that abbreviated as VIP. Okay, so that is called the VIP is otherwise called as the vasoactive intestinal peptide. Okay, it's called as vasoactive intestinal peptide. Okay, so this vasoactive intestinal peptide causes what? Vasodilation. So remember, it is not the acetylcholine that is causing the vasodilation, but another substance, that is the vasoactive intestinal, that is a co-transmitter that is secreted along with acetylcholine only over here. Okay, only in the GI we can see them, the vasoactive intestinal peptide. Okay, whereas the sympathetic causes vasoconstriction, whereas this vasoactive intestinal peptide that is released co-transmitted with acetylcholine causes vasodilation. So vasodilation also helps to secrete watery saliva. Okay, so that is how the major regulation of the salivary secretion is happening. Okay, so let's understand these same principles of the regulation through a question. Okay, so let me show you one question over here just to discuss, okay, about the, just to make sure you understood this. Okay, so over here it's a question that is talking about like a pathological condition and then asking you about the regulation. So let's see what is the pathological condition. The name of the pathological condition is already given in the question itself. It's called as the Sjogren's syndrome. Okay, so let's see what are the features of the Sjogren's syndrome. So a 56-year-old female is seen with dry mouth. Okay, so this is the number one symptom for the Sjogren's syndrome. Okay, the dry mouth, and also dry eyes. Okay, so dry eyes and dry mouth. Okay, so this is the second symptom. Okay, so it's like have dry mouth, okay, and the dry eyes makes it like a scratchy eyes. Okay, so the question is describing it as a scratchy eye. So why it became scratchy? Because of the dryness. So she complains of scratchy eyes and that her dry mouth makes it difficult for her to eat. Okay, and she has the history of bilateral sialadenitis. Okay, sometimes sialadenitis will be with stone. Most probably it will be with the stone. So in this case, this adenitis, the inflammation has happened for the glands is without the stone formation. Okay, so and that resolved without treatment. Okay, so which is when her dry mouth symptoms started. Okay, so this is like a stimulus that has caused the Sjogren's syndrome. Sjogren's syndrome is actually a autoimmune disorder in which the SSA and SSB antibodies are causing the damage to the salivary glands. Okay, so the question is saying she is positive for SSA and SSB antibodies and is diagnosed with Sjogren's syndrome, which causes the autoimmune destruction of the salivary glands. Now she is given artificial tears and her for her dry eyes and artificial saliva for dry mouth. So that's the treatment approach. So that is the vignette part. Okay, all this is the vignette part, not the question part. Now the question is the physiology part. Okay, so which of the following is the main form of cellular regulation in the cells that have been lost to this disease? Okay, so the cells that is being lost to this disease is the salivary SNR cells that can cause the secretions. Okay, so what is the major regulation? Just now we have seen that is the acetylcholine. So remember, we have mentioned about the aldosterone, right? Okay, so but in this scenario the aldosterone is not the correct option, because aldosterone can influence the sodium and the potassium concentration of the saliva. Okay, what does it do to the salivary sodium concentration? It decreases the sodium concentration and increases the potassium concentration in saliva, but that is only the influence by acting on the ducts, but not controlling the or regulating the secretion of the saliva. So acetylcholine from the parasympathetic nerve is the one that causes. Okay, so all these like motilin, secretin and cholecystokinin, all these are the hormones that is there in the GI tract. We are going to study about those like hormones later. Okay, so we have motilin, we have secretin, we have CCK or the cholecystokinin. All of them are the hormones of the GI tract, but these hormones including the does not have an influence on the salivary secretion. So the major secretion regulator of the secretion is acetylcholine. Okay, so in the next video we will continue with the next process, that is the mastication or the chewing. Okay, thank you.